Mic test. Hopefully, through chat, can you verify that you can hear me? Hindi ko kayo marinig. Okay, so I'll just continue. So we're familiar with the Gram-Stein procedure, right? So uh, the gram positive will be purple colored, and then the gram negative will be red or pink. Okay. So the first step in gram stain procedure, you add the primary stain, which is the crystal violet. Okay. So all the cells, whether all the bacteria, whether gram positive or gram negative, will have the crystal, will have the violet color or the purple color. Okay. So the gram positive will have the thicker peptidoglycan cell wall, and the gram negative will have the thinner peptidoglycan cell wall. Both of them will absorb. And then you add iodine. So this more than, or it acts as a, it makes a complex with crystal violet that helps the crystal violet bind more. And it makes the dye less soluble, so it makes it adhere to the cell wall. Okay, so both are still purple or violet or blue. So when you add alcohol, so the alcohol washes away the purple or the crystal violet that's not binded well. So for those with thick peptidoglycan layers, the the color still remains. So the gram-positive cells will remain purple, while the gram-negative bacteria will not retain the color because of their thin peptidoglycan layer. So now that you have removed the dye from the gram-negative bacteria, so you add the counter stain. So in this case, you add the saponin. So this will give a pink or red color to the gram-negative bacteria. Okay, so okay, so sometimes there are errors, no, in interpreting the results of Gram-Stein because sometimes you have older bacterial cells. So when that happens, because of the damage of their cell walls, it may appear the gram-negative sila, but it's actually because of their uh, old bacterial cells. So you have to analyze. And then sometimes you might add too much decolorizer. Okay, so you might not wash away the decolorizer. It will even for gram positive. If you leave the decolorizer too long, you can remove the crystal violet dye even in gram positive bacteria. Okay, so you must be uh, careful. Okay, so you see here the gram positive bacteria, that's the purple colored. Then the gram negative, ayan yung nasa background. So you have the pink color. So yung gram positive, Staphylococcus aureus, and then you have the uh, Escherichia coli. Yun yung pink. Okay, so this is very helpful. It helps the clinician classify bacterial pathogens and associated specific properties. So dun sa categories natin ng bacteria, kaya discuss ko to sa lecture because it's important dun sa discussion natin. Okay? So your gram-negative bacteria, because of their structure, it tends to be more resistant to antibiotics than gram-positive bacteria. Okay? So yeah, gram-positive. So another way is the acid-fast stain. So depende, kasi may mga ibang uh, mga organisms, because of the presence of some chemicals in their cell walls, hindi nag-absorb. Okay? Uh, so you need acid-fast staining to be able to differentiate the types of gram-positive cells. Those that have waxy mycolic acids in their cell walls and those who do not. So you have the Ziehl-Neelsen technique and the Konyon technique. So in this, both of them use carbofluoxetine as the primary stain. Okay. So dun sa may waxy mycolic acid, so they retain the carbofluoxetine even if you add the acid alcohol solution. So the acid alcohol solution acts as a decolorizing agent. So you have the methylene blue as the secondary counter stain. Okay, so non-acid pastels are stained blue. So in the Ziehl-Neelsen method, it uses heat. So this will infuse the carbofluoxetine into the cells. In the Kinyon method, it does not use heat. Okay, so they are techniques because if you want to study some diseases like Mycobacterium tuberculosis, so you want to confirm the presence of this bacteria, acid-fast bacteria. So these techniques are used. Okay, so yung Mycobacterium tuberculosis, nakikita through the Ziehl-Neelsen staining method. Okay, so there are organisms that instead of the cell walls, you have capsules. So they have you can use the presence of the capsule to absorb the dye. Okay, and then these capsules also protects the organism. No? So yung certain types of bacteria na may capsule, mas mahirap patayin. Okay, so it is related to the ability of the organism to cause disease. Siyempre, kung mas mahirap patayin, mas magkakause ng disease yan. Unlike without the capsule, it's easily destroyed by our immune system. Okay? So, because capsules do not absorb most dyes, then use a negative staining technique. So this is used for capsule staining. Okay, so you can use the negative staining to stain the background, and then the positive stain to make the organism absorb. So the positive stain colors the body of the cell, while the negative stain colors the background. So you have a halo because the capsule does not absorb. Okay? So kita nyo dyan sa Cryptococcus neoformans, you have the halo surrounding the cells. Okay? So mas prominent dito sa isang picture. No? So sa right picture, you use crystal violet. Use crystal violet and copper sulfate dyes. They cannot penetrate the cells, but you color the background. Okay? So may halo, kita mo yung white color. That's the capsule. Okay. So we also learned na yung mga bacteria, they can protect themselves during times na low resources or harsh conditions. So they create endospores. So we can use this, we need to, we want to find out if there are endospores in the sample. So I use the endospore staining techniques. So in this technique, you use two stains to differentiate endospores from the rest of the cells. So the most used endospore technique is the Schaefer-Fulton method. So in this method, you're going to use malachite clean into the endospore. Okay. So washing with water decolorizes the cell, but the endospore retains the green state. And then the cell is then counterstained pink with saffron. So using this technique, you can be able to locate the endospores or the shape of the endospores. So in the use of that technique, the green color will appear. So it may appear within the vegetative cells or a separate. Okay. So if there are no green color, so there are no endospores present, then only the pink will be visible. So you use this technique to identify Bacillus and Clostridium. So these are bacteria that produce endospores. Okay, so they're important clinically. So dito sa slide na to, you can see the green endospores within the vegetative bacterial cells. Okay. And then, of course, we also want to look at the flagella. So we already studied this. Bacteria have it, eukaryotes have it. So they cannot be seen typically because they're very thin. So you need the staining technique that is specialized for flagella. So what you want to do is you want to make the flagella more thick. So you apply a coat. So this is a mordant. So this coats the flagella, and then you add rosanilin, rosanilin, and or you can use basic fixing. Okay, so in this technique, kitang-kita yung yung flagella ng Bacillus cereus. Okay? So that summarizes the basic stains. So yung mga specific dyes, methylene blue, crystal violet, malachite green, basic fuchsin, carbofuchsin, and safranin. So these are basic dyes. So you want to have the structure absorb the dye. So this is the positive stain in contrast to the negative charged cell wall. So like forces or like charges attract. So yun, absorb. For acidic stain, you have using acid fuchsin, rose bengal, and Congo red. So again, the stain positively charged molecules and structures such as proteins. Okay, so it isn't absorbed by the cell wall. So this is the background. So you can use depende rin sa structure ng mismong cell, no? So you need to understand the cell's chemistry. Kung mag-repel siya, magiging negative stain siya. So the third one is the negative stain. Ito, ano ka talaga, you're after the background. So the cell, you're sure that the cell will not absorb the dye. So yeah, NJ engrossing. So it stains the background. So you have a dark background with lights present. Okay, so yan yung mga summary. So gram stain, we discussed that already. Acid stain, acid fast stain, so sa mga, lalo sa mga mycobacterium, may folic acid. So you use to distinguish acid fast bacteria. So acid fast bacteria are red, and non-acid fast cells are blue. Yung endospores, you use heat to stain the endospores, so the technique is Schaefer-Fulton. So you use malachite green. It is used to distinguish organisms with endospores from those without. So you want to know the presence, of course, of the endospores. And then for flagella stain, you need to coat the flagella to make it thicker. So use tannic acid or potassium alum. And then you can use pararosaline, rosaline, and then you have the basic fuchsin. This is if you want to view the flagella. Okay. And then the last one is the capsule. So you want a halo. The capsule doesn't absorb, so you have a halo surrounding the organism. So you use negative staining with NJ or degrossing. So you have the halo because the capsule will not absorb. So the counter stain can be used to stain the cell and leave the capsule clear. So it's used to distinguish cells with capsules and those without. Okay. Tapos, ayun, yung parts of the cell. Basically, picture, I'll be showing a picture or pwede rin mismong slide. So, uh, I will ask you anong part yun. Okay? So, in contrast to multiple choice, wala masyadong picture, more on function. Sa practical exam, I can show you the picture. So sabihin nyo, point where the mitochondria is. So yun yung mga pwedeng tanong. Okay? So I will get an immediate answer. So random yung questions, no? So may set ka ng questions. And siguro per person, hindi naman siguro abot ng 10 minutes. We have 5 hours naman. So I can ask all of you by the time the session is through. Okay?
What did you do?
What else? So yun yung coverage. So again, the coverage will be, let's see. Lagay ko na sa chat. So the coverage will be about the parts of the microscope, how to use the microscope. Okay? So staining techniques, yung kaka-discuss ko. Tapos yung cell parts. And then siguro yung last yung how to prepare microscope slides. So yun lang. Tapos yung mga pictures din. So you have to be familiar with yung yung nasa slides, di ba? Yung mga ball box, yung diatoms. So familiar dapat yun. So I'll show you a picture and then you will be able to know what that organism is. Okay? If you have any questions, please lagay niyo sa chat kasi hindi ko naririnig yung hindi ko kayo naririnig. So, babasahin ko na sa chat. Ilang points yung practical exam? So, siguro 5 questions. 5 or demonstrations. Mabilis lang naman. So, over 100. Tapos 5 questions. So, ano naman, may rubrics naman. Okay? Okay, for the last part, yung preparing konting discussion dito sa preparing microscope slides. Ayaw mo gusto. Yeah, sorry. Ayaw. Ayaw gusto. Professional. So, konti lang yung coverage nung ano niya, sa lab. No? So, ito yung, so first will be the parts of the microscope, how to use the microscope, and then yung staining techniques. So, lahat dyan sa second PowerPoint. Okay? And then, ito lang, ay yung mga pictures din. Pictures ng mga different organisms. And then ito last, no? How to prepare a slide. So you can select a clean slide, dapat walang fingerprint. So importante, bakit mo kailangan ng, siyempre ayaw mo maka-fingerprint yung slide mo kasi lalabas yun sa under the microscope. So that's why it's important na hawakan sa side. Okay, huwag na hawakan yung mismo slide and you use gloves. Okay, and you use tweezers. So pag contaminate may fingerprints yan, you clean it with liquid soap or water or wipe it with alcohol. So don't use paper. So inspect the specimen if it needs to be sliced. So dapat maliit enough. Tsaka dapat translucent. Okay, so hindi mo pwedeng makita sa microscope ng bato, ganun. So ang pwede dyan mga wings ng insect, plant leaf, gano'n. So you can use that for dry mount. Pwedeng hair, pwedeng fur. Okay? So yan, mga dry specimen. Use forceps or tweezers and lay it on the side of the slide. Then use cover slip to prevent the specimen from tumbling off the slide. Okay? So, alam niyo naman yung itsura ng cover slip. And then yung wet mount. Okay, so babasahin nyo. So, may water droplets ka, eyedropper, to drop one water into the exact center of the flat or concave slide. So, kaya nga wet mount. Okay? So, sa plant leaf, for example, you can use nail polish instead of water. Kung permanent yung gusto mong gawing specimen. Okay? So, use a razor blade or toothpick to cut out or scrape small amount of your wet specimen. So, yung ginamit natin yung cheek cells. To place the specimen in your drop of water. And then you put the cover slip on top of the slide. So, it's important na hindi magkakaroon ng bubble. Makikita kasi yun sa microscope. So, dapat you hold the cover slip at 45 degrees. Set one of the edges down next to the specimen. So slowly lower. So, ang mangyayari dyan kasi kung di maayos yung pagkalagay nyo, magkakaroon ng bubbles. Baka isipin nyo part of the specimen. Pero actually, it's air bubbles. Okay, so slowly lower your cover slip. You can use a paper towel to absorb the excess liquid. And then yung ginamit nating dye for this, we use methylene blue for the first experiment. Okay. So iodine will make you see the plant cell. Sa ginawa natin sa onion. So wait till while the staining agent is drawn under the cover slip. So mag-slip under. So it may take 5 minutes. And wipe the excess with paper towel. Okay. So yan. So yun lang yung ano natin. That will be the coverage of our lab. So for next meeting, wala muna. As requested, we will have the exam muna. So next week, that will be April. Okay, siguro